Welcome to Learner's Planet. Now again we are starting this next session of the chapter Fundamental Unit of Life. Till now, or in the previous sessions, we have actually studied about the basic properties of the cell, as well as we have started with the study of various parts of the cell. We have completed with the plasma membrane, and we have studied how it is actually behaving as a boundary for the cell. Besides, we have studied that in plant cells and some other kind of cells, how the another layer is needed. So this particular plasma membrane is covered by another membrane known as cell wall. Besides that, we have studied about the nucleus in the eukaryotic cells, as well as we have studied about the cytoplasm. Now in the previous session, we have ended up with the structure of nucleus in a eukaryotic cell, and now we are continuing with the same topic in this particular session. So here, now we are studying the nucleus in the prokaryotic cell. You remember what is meant by the prokaryotic and the eukaryotic? Eukaryote, it means that the cells which are having true type of nucleus. Now which nucleus is known as the true type of nucleus? So it is the one which is having the membrane, which is surrounded by the nuclear membrane. This nuclear membrane separates the cytoplasm from the nucleoplasm. So in a eukaryotic cell, the two bodies, that is the organelles or the cytoplasmic region, is completely separated from the nucleoplasm. Why? It is because of the presence of a nuclear membrane. Besides this, the nucleus in the eukaryotic cell is having the distinct region, a particular dense area inside that we call as nucleolus. The nucleolus is also known as a factory of ribosomes. The reason is because it is involved in the formation of ribosomes. Ribosomes, you remember, it is involved in the synthesis of the proteins. Besides, it also contains the RNA as well as the proteins over there. But this distinct region, that is nucleolus, is absolutely absent in the case of prokaryotic cell. So besides this, in the case of eukaryotic cell, in the eukaryotic nucleus, we have also seen that there are chromosomes which may be single, or it may be more than a single or more than one. Whereas in the case of prokaryotic cell, again they possess only a single DNA, and that may be either circular or it is linear. In the case of eukaryotic cell, generally it is linear. In some unicellular organisms, it may be circular, but in general it is always linear. But in prokaryotes, it is most of the time it is circular in nature and always single. That is more important here. Besides this, in the case of nucleus of eukaryotic cell, there is a presence of nucleoplasm. That means the matrix inside the nuclear membrane, which is holding the particular region, nucleolus, or the nuclear material, that is chromosomes, etc. That matrix is known as nucleoplasm. But in case of prokaryotes, no such separate special matrix is present, or it is found in which the nuclear material is present. Rather, here, as the nuclear membrane is completely absent. So you can imagine here that in this case, the nucleoplasm is having no particular identity. Here, the region is actually intermixed with the cytoplasm. So in the case of prokaryotic cells, the region in which the chromosome is actually present, that region is known as nucleoid. That means there is no separate membrane, there is no separate matrix, but the chromosome is of course present. That means the controlling center is definitely present in the case of prokaryotic cell also. And chromosome, this type of chromosome which is present in a particular region in a prokaryotic cell is known as nucleoid. Now such type of prokaryotic cells, they not only have this type of primitive structure in the name of nucleus, but there is another thing also, and that is they also lack membrane-bound organelles. All the membrane-bound organelles, they all are absent in case of prokaryotic cells. Can you think of certain organelles like endoplasmic reticulum? Like we can say, for example, mitochondria, Golgi apparatus. There are so many organelles which are usually absent in the case of prokaryotic cells. But still, the function of each organelle is performed by the cell, prokaryotic cell, with the help of other structures, with the help of such primitive type of structures itself, like its own cell membrane, like its own cytoplasmic inclusions, like vacuoles, and so on. So all these functions are performed by the various parts of the cells, but the true type of organelles as well as a true type of nucleus is absent. So this is the reason why it is called as a prokaryotic cell. The word pro means primitive, and we are relating it with the nucleus because the nucleus is also a membrane-bound organelle which is present in the eukaryotic cell in a developed form, but its modified primitive type of form is present in prokaryotic cell also. The other membrane-bound organelles, of course, they are absent. So these cells which contain primitive type of nucleus, they would be termed as prokaryotic cells. We can also clarify these facts with the help of this particular diagram. This is the structure of a bacteria. In this case, you can see the various parts like plasma membrane, flagellum, cytoplasm, ribosomes, and so on. So each particular part is having its own special function. Plasma membrane is the outer boundary, but this outer boundary is covered by two different layers further, and those are cell wall and capsule. So these two structures basically protect the cell from the surrounding conditions. Bacteria is a unicellular organism, and if it is unicellular, you can imagine the minor changes in the surroundings can cause harm to such a structure, but they have a capacity to withhold such type of conditions. So can you imagine why is it possible? It is possible due to the presence of these extra membrane in its surrounding, as well as the functional chromosome, functional genetic material. Now this genetic material, it is present within the cell in the cytoplasm, and such type of DNA or the chromosome which is devoid of nuclear membrane, it is known as nucleoid. So functionally it is a DNA only. We have already studied that these genetic material, they are chromosomes. We can say like they are actually the nuclear material. In the case of bacteria also, it is chromosome. These chromosomes, they are having genes located over itself. Now these genes, they are the functional hereditary unit, right? They are coding for the particular function or the characteristic. Besides these functional units, they are chemically made up of DNA. You remember the full form of DNA? It is deoxyribonucleic acid. Now these deoxyribo, it means what? They are having the type of deoxyribose sugar in it. Further, the DNA is a long chain double helix structure. And this double helix structure means what? The structure seems to be something like this. This is a one strand of DNA, and this is the another strand of DNA which is coiled each one above the other. So this type of DNA, it is having the small units located over itself. A single chain is a polymer of small units, and these small units, they are known as what? They are known as nucleotide. So each DNA is made up of what? It is made up of nucleotide. Now these nucleotides, they are made up of three units. What are those 3 units? We have already studied in the previous session. They are sugar. Which sugar is present in DNA? It is deoxyribonucleic acid. I'm sorry, the sugar is deoxyribose. Whereas the other part of this nucleotide is a base and an attached phosphate group. Now how these are located? The phosphate group is located towards the outside of this particular chain. Then this backbone, this strand, is actually made due to the binding of sugar units. And towards the interior of the helix, in this particular gap, they are having the base inside. These bases are the nuclear bases. They could be of different kind, like adenine, thymine, then we can say like cytosine and guanine. So these are present in the middle of this particular section. Further, they are forming a bond with each other. The bonding between the two parts, the two bases of each strand, they will help the helix to get attached with each other. They will help in maintaining the helix structure of the DNA. So you can see here now that although the nuclear structure, nuclear complexity is different in the case of prokaryotes and the eukaryotes, but still the backbone, the nuclear material structure is same in the case of each cell, whether it is nucleoid or it is a developed nucleus. In each case, the genetic material is supposed to be the chromosome. And in that, of course, it is meant for storing the functional units called genes. So these are actually present in the center region and they are not having a distinct membrane. So they are present within the cytoplasm only. So such a nucleus is known as what? It is known as nucleoid. Besides this, the other parts are also present like ribosomes. In the case of eukaryotic cell, how the ribosomes are present? They are attached over the endoplasmic reticulum. Whereas in the case of bacteria or a prokaryotic cell, these ribosomes are scattered freely or they are present freely inside the cytoplasm. The function is of course same as that in the case of eukaryotic cell, and that is what? That is protein synthesis. Besides, in the case of these prokaryotic cells, all other membrane bound organelles are absent. So those functions are generally performed by the plasma membrane itself. Let us consider an example like mitochondria. What is the function of mitochondria? It is a site of respiration. It is involved in the synthesis of energy-rich molecules. So if mitochondria is not present in the case of prokaryotic cell, then what? Then it is performed by the plasma membrane itself. So in the prokaryotic cell, this plasma membrane is forming certain invaginations. They are showing certain folds towards the interior of the cytoplasm, towards the inner side of the cell. And these folds are known as mesosomes. Now these mesosomes, as the membrane gets folded and forms a mesosome, the surface area has been increased. Due to this, more and more oxidation or more and more cellular respiration can take place. So this is how certain modified structures are present in the prokaryotic cells so as to carry out the basic life-sustaining functions. Besides this, they are also having a bacterial flagellum, but this is not necessary that each prokaryotic cell is going to possess this flagellum. But if flagellum is not there, there might be certain other kind of structure. Usually flagella or minute villi, or we can say like cilia-like structures are present, and such extensions actually help in the movement of the organism. They help in the locomotion of such organisms. So now you can compare that not only the nucleus is changed, it is not only nucleus which is different in prokaryotic cell, but such difference also affects the other parts of the cell also. All other functions would be affected due to such type of primitive kind of nucleus. Further, the important thing is that although all these changes are present within the cell, but all will be controlled by the chromosome itself. So here nucleus is performing as a brain of the cell, or it is a controlling center of the cell. Now let us study the basic functions of the nucleus. Let us compare it in the case of prokaryotes as well as in the eukaryotes. As we are saying that nucleus or nucleoid, it is a brain or the control unit of the cell. So functions are more or less similar. Let us study one by one each function of a nucleus. The very first function of this nucleus is that it stores the information code. Do you understand what we mean by this particular point? It stores information code. Information about what? Information about all the functions of the body. Now how these are actually encoded? We described in the previous slide that chromosomes, they locate the genes. Genes are located over there, and the genes are known as the functional unit of inheritance. Why so? Because it is called as this information code. So information code is basically a gene which is actually coding for the particular function. Now how this gene is actually coding for a function? So this is because it is a code for a particular protein, right? These genes they are chemically DNA. These DNA, they gets converted into the RNA, that is ribonucleic acid. It is another form of a nucleic acid. Deoxyribonucleic acid, it is a unit in which there is a presence of deoxyribose sugar. Another one we are saying is RNA. This RNA contains ribose sugar. So genes which are a DNA, they convert into the RNA. Then RNA, they are translated into another form, and that is known as what? It is known as proteins. So these proteins are actually coded, or they are formed by the genes only. Although these are the stepwise process, but the final product which is produced by the gene is in the form of a protein. Now can you think another thing, that if genes are able to produce only the proteins, then how the other chemicals or other functions of the body are controlled? Because a function or the living organism is comprised of many different kind of biomolecules. So if only protein is produced by genes, then how those other functional molecules are produced? So the reason is that, or we can explain it with the help of another function here, another point. These proteins which are encoded by genes or which are produced by the genes, now they can function as different things, like they can function as an enzyme or hormone, transporters, regulators, structural proteins, etc. They can be actually functioning in a different ways in a body. Either they themselves are a functional product. Let us say, for example, hemoglobin. In a hemoglobin, globin is a part which is a protein. Then this protein is attached with another moiety called heme. So if the globin is attached with the heme part or the iron part, then it is known as a hemoglobin. So in this case, the globin, a protein which is produced by the gene, it is directly functioning as a functional molecule. The protein itself is a functional molecule here. Further, this hemoglobin is needed for what purpose? It is needed for the, you can say, oxygen carrying, it is functioning as an oxygen carrying molecule. So it helps in the transportation of this respiratory molecule. Besides this, there are so many examples of similar type of proteins. These particular proteins, they can function as an enzyme. Enzymes, they are the biocatalyst. Now what you mean by biocatalyst? In chemistry, what is the function of a catalyst? Catalyst basically accelerates the rate of reaction. If the catalyst is added, then it will increase the rate of reaction. The same function in a living system is performed by the enzyme. So the enzyme is called as a biocatalyst. So these enzymes, they will help to carry out the all different kind of chemical reactions in a living system. So how it is actually helping? Whatever molecule is synthesized in the body, they need the substrate which is provided as a raw material either from the diet or by the body itself. Now what is needed is, we need certain agents which can help to increase the rate of reaction, to increase the rate of production of a specific molecules, and that function is performed by the enzyme. Enzyme is chemically a protein, and protein is actually coded by the gene. So you can see the direct involvement of this, the function of protein as an enzyme now. Similarly, hormones. The hormones, either there are certain type of hormones which are chemically proteins. They are known as peptide hormones. Now these hormones, they are also, if they are proteins, then definitely they are synthesized by the genes itself. Not only this, the other hormones which are synthesized in the body, they need the enzymes so as to be produced. So enzymes, again they are the proteins. Thus you can see the involvement here again. Then the transporters. You remember where we have studied these transporters? We have studied it in the plasma membrane. There are certain channel proteins, carrier proteins in the plasma membrane, right? Not only in the plasma membrane, but any other cell membrane. These carrier molecules or the transporters are present, which help in the movement of material in and out of the cell in a controlled manner, of course. So these are chemically what? Again, a protein. So transporters, they can be coded by the genes now. Further, they can function as the regulators. What do you mean by regulator? Regulator means they are controlling the rate of the reaction. As we have said, enzymes. These enzymes, they accelerate the reaction. Now just imagine, if the reaction is taking place and it continues to take place in an uncontrolled manner, then what will happen? The product which is formed will start accumulating. The normal level is going to increase. So this should not happen. These enzymes, they should accelerate the reaction, but the control should be there, that how much product is needed. So once the required amount is being synthesized, the reaction has to be stopped. So, or in another way we can say, these enzymes, they may be in the inactive state. So when the substrates are available, the enzymes have to be activated. So there are certain other molecules which will help in the activation of these enzymes. Then once activated, they will accelerate the reaction. So such molecules which control the functioning of these enzymes, they are known as regulators. So these regulators, they can function positively or negatively. The one which is functioning positively is called as an activator. The other which is functioning negatively is known as inhibitor. So this way, almost all of the functions of the body, they are controlled by these regulators. Now most of the regulators in the body, they are chemically proteins. So again you can see the direct link of genes with the regulators. Besides that, most of the proteins, they can function as a structural protein. Can you think of any example of a structural protein? The proteins which are present in the plasma membrane, it helps in giving the structure to the cell. You can say like keratin, it maintains the structure of your hairs. There are so many functions, there are so many proteins which help in the maintenance of a structure of the cell. And such functional proteins are known as structural proteins. So there are many roles of the proteins in the body, and either directly or indirectly. And thus we are saying that these proteins are formed by the genes and is an important function of a nucleus. Besides that, another function is it controls all the metabolic activities of the cell. Why it is so? First of all, what do you mean by the metabolic activity? So metabolic activity or metabolism means the set of all biochemical reactions. Now code for each such type of reaction is basically encoded by the genes only. Like we can say which substrate is needed, which enzyme is needed. So all these enzymes, the substrates and the regulators all are secreted by the genes only. So thus we are saying they are controlling all of the metabolic reaction of the body. Further, they can help in the regulation of the cell cycle. What do we mean by this point now? It means, you know what is cell cycle? Cell is continuously dividing in the body, of course in a controlled manner, right? Cell cycle means, suppose there is a cell in the body, when it matures, it will undergo the cellular reproduction. That means it will divide. Or why this cell division is taking place? It is having basic functions like repair. So if some cell is actually injured, it has to be replaced by the new cell, because that repairment has to be removed from the body. So to control for the normal functioning of the body, cell has to divide. They have to take up the function of those injured cells. Similarly, we can say it is for the sake of replacement. Like if there are some old cells in the body and they have crossed the aging process. The age is so high that the rate of its function has been declined. So such cells, they have to be removed. They will undergo the cell death. Now if all the cell will die in the body, then who is going to perform the function, basic function of the system? That is actually performed by the new cells which are replacing it. And from where these new cells are coming, it is by the cell division. So each cell will undergo the cell cycle. A cell will mature, it will divide. Then further again it will re-enter into the cellular cell division phase. So this cell cycle or this cell division, it should take place in a controlled manner. Why it is so? Else you imagine, if the cells are continuously dividing in the body beyond the need of the body, then what will happen? They will start accumulating. So the entire function would be disrupted in the body. Entire structure will be disrupted. We can suffer from various kinds of diseases like tumor. We can develop the tumor in the body or the cancer. So such situation should not arise, and hence the rate of cell division should be actually controlled. And this control is a function of the nucleus. The nucleus, it contains the nuclear material, and this nuclear material basically gives the command to control the cell division cycle. And thus we can see there is a direct involvement. Why so, or how it is so? It is because they can produce the enzymes, they can produce the transporters, they can produce the regulators. So these enzymes, regulators together control that whether the cycle has to be accelerated or it has to be inhibited. So these nucleus are basically controlling the rate of cell division cycle. Beside all these functions, the important function is the transformation or transmission of these particular information codes from one generation to the next. So not only the cellular reproduction is controlled, it also helps in the transfer of a particular gene or the characteristics from parental generation to the daughter or the offspring generation. So these codes or these information are actually inherited in the form of genes only. So this is why we say that the nucleus is actually control center of the cell. It is known as a brain because it can control all the metabolic functions. It is controlled as a director, as it can control the metabolic functions as well as it can regulate the cell cycle also. So these are certain basic functions of the nucleus. Further, we will continue with the basic other points related to the structure of the cell. Till now this much, and thank you so much. We'll continue in the next session.